Good afternoon, or today we are going to see WHO guidelines for evaluation of finished herbal products. WHO guideline includes assessment of quality, the quality of crude drug material, then plant preparation and finished products. Assessment of stability. The stability is related to the shelf life. Assessment of safety. Documentation of safety based on the experience and toxicological study. That means toxicological data and how the drug is safe or what are the requirements to prove the drug it is not toxic. Assessment of efficacy by ethnomedicinal information and biological activity. Ethnomedicinal information, what is the use or what is the pharmacological activity of that drug and biological activity of that drug. There are three pillars for the herbal drug and their rational use, that is quality, safety and efficacy. Assessment of quality. Quality assessment is done for the crude drug plant material, that means only the crude drug part or part of the plant material which is in a dried form or in a processed form. Then drug plant preparation or extract, that means the prepared product of the drug. It may include the extract, volatile oil, fixed oil, gum, etc. Finished product. The finished product consists of the plant material or plant preparation along with the excipient. First, we will see the assessment of quality of the crude plant material. In that, identity of the drug, that means botanical identity, which part is used, then what is the biological source, then authentication of that part or drug. Physicochemical characteristic, which include chromatographic profile, ash value, extractive value, refractive index, polarimetric reading, moisture content, volatile oil. Pharmacological parameters, which include biological activity profile, that means what is the activity of that drug, bitterness value, hemolytic index, then astringency, swelling factor, foaming index, etc. Toxicity detail of the drug, which include heavy metal content and pesticide residue. Next, the plant preparation or extract. Assessment of quality of plant preparation or extract. Botanical extract monograph, that means which solvent is used, what is the ratio of the solvent and the drug, and what is the content of the drug, what is the content of active ingredient in the plant part or drug. Physical analysis, melting point, viscosity, density, optical density, specific gravity, percentage yield, extractive value. Extractive value is related to the alcohol soluble extractive value and water soluble extractive value. Then moisture content. Chemical analysis of plant preparation or extract. Qualitative chemical test, which include identification of phytoconstituent, that means which type of secondary metabolite is present in a plant extract. Quantitative analysis, which include acid value, iodine value, ester value, saponification value. Heavy metal and pesticide content also include the chromatographic fingerprinting. Spectral analysis, which include UV, IR, NMR and mass. In UV, lambda max or how many number of components are present in a extract that can be identified. Functional group, to detect the functional group by IR spectroscopy. Mass fragmentation and mass spectra and identification of compound by the NMR. Finished product. Detailed description of the finished product. That means which manufacturing procedure is used, what is the formula, what is the content, what is the quantity of that particular drug or excipient in the given product, including the amount of excipient, finished product specification, method of identification and quantification. Which method is used for the identification. Then B, that is a parameter for particular dosage form. Finished product is prepared in which dosage form. That dosage form parameters are essential to perform to identify the quality of that product. For example, if tablet is there, then shape, appearance, disintegration test, dissolution test, friability test, etc. should be determined. In case of emulsion, flocculation sedimentation rate, dilution test, conductivity test, globule test, pH, viscosity, etc. should be tested. In case of creams, particle size, extrudability, spreadability, viscosity, pH, etc. And last but not least, that is a for imported finished product, confirmation of the regulatory status in the country of origin should be required. If the product is imported, then regulatory status of that country, and if the product is prepared in India, then what are the regulatory status or regulatory requirements related to that product in India should be checked or passed for the particular finished product. So this is all about the assessment of quality of crude plant material, drug plant preparation or extract, and finished product. That is the only first parameter of the WHO guideline for finished herbal products. Remaining parameter we will see in the next lecture.